So I'd, of course, like to thank the organizers of this meeting for the opportunity to come here. This is a wonderful location, and I will have to go back to the United States and ask the people at Nescent to please improve their decor, because I'm afraid that they're running a little bit behind their counterparts. I feel a little funny giving this talk at a conference on speciation, because I'm going to tell you about not speciating. And the motivation for this comes from something that is a classic in the speciation literature. I suspect probably everybody in the room is familiar with the benthic and limnetic species pairs of three-spined stickleback that are found in, give or take, about 6 lakes in British Columbia. And these Sympatric species pairs are repeated in several lakes, and in each of those lakes you see a nice bimodal distribution of phenotypes. You see genetic differences between the populations, behavioral differences, ecological differences. And so these populations really seem to represent fairly good biological species, although unfortunately it is possible for environmental change to undermine the reproductive isolation, and so we've seen the loss of at least one and possibly more species pairs. Now, the wonderful work that Dolf Schluter and his colleagues have done on this system sometimes leads us to believe that stickleback speciate, and that they do so quite easily. But I think it's important to remember that there are also thousands of lakes where, to the best of our knowledge, there are only solitary populations of stickleback. Where, for example, in Cranby Lake, which is right next to Paxton Lake, you see a unimodal distribution. Many times we see an even more normal distribution than shown here. And to the best of our knowledge, these thousands of populations represent panmictic demes that, although one lake may be different from another, and bigger lakes maybe look more limnetic and smaller lakes look more benthic on average, we still seem to have many instances in which there are only solitary species. And so the question that I'm going to pose is, why do most populations seem stuck here rather than here, given that the existence of species pairs proves to us that it is possible for lakes to support and maintain bimodal trait distributions and reproductively isolated sympatric populations. Now, a quick note, I should point out that to the best of our knowledge, these species pairs were not necessarily derived sympatrically. There's some evidence there may have been a double invasion scenario followed by ecological character displacement and reinforcement, although the evidence for double invasion is not completely straightforward either. And so there's some ambiguity, but there's no particular reason to suspect that these were sympatrically derived sympatric species pairs. So I'm going to lay out for you the argument that these solitary populations do in fact exhibit many of the features that we've heard about in the past day that define the process of sympatric speciation. Namely, there's ecological variation related to traits that are subject to disruptive selection, and those same or correlated traits are the basis for assortative mating. So all 3 of these patterns and processes can be observed in the solitary populations, which suggests that these preconditions for sympatric speciation are indeed met. So then why not speciate? So to begin with the ecological variation, the benthic and limnitic species pairs are so called because the benthic species predominantly feeds on the benthos, either on mud or rocks or vegetation, eating macroinvertebrates, whereas the limnetic species feeds more in the open water. So a master student of mine, Kim Hendricks, simply spent 8 hours a day in 12-degree water snorkeling, watching fish that we had individually marked with colored beads, and asked, what fraction of time is this individual fish executing various feeding behaviors, feeding on the surface, in the midwater, or on the benthos? She did this over a course of 14 days on 30 marked individuals and recorded an average of 165 feeding attacks by each individual fish. So we can summarize the data in a ternary plot. This is representing the proportion of strikes at the benthos, surface, and midwater, and any dot is an individual that summarizes the proportions spent on each of these three activities. So if you're over in this corner here, you're doing 100% benthic strikes. And you see there are a fair number of individuals that are pretty much always feeding on the benthos. But there are individuals who are 70% of the time feeding in midwater, or 70% of the time feeding on the water surface. And we have strongly statistically significant differences between individuals. If we break this down day by day for each individual, we see a great deal of consistency, repeatability by individuals. Now, these differences in where individuals are feeding are despite the fact that all of these fish were constrained inside a net. This net contained 30 meters of shoreline, and so they're in a fairly small, constrained space. Now, these different microhabitats present different food types, so it's probably not surprising that if you feed in different places, you eat different things. We can summarize that using stable isotope analyses. Carbon gives us a measure of how benthic or limnetic your prey are, and individual stickleback, you can ignore the shaded and unshaded dots for today, range from being nearly 100% limnetic to 100% benthic based on their carbon signatures. If you look at the nitrogen axis, it represents trophic position, and we see some variation in trophic position as well that's explainable because limnetic prey tend to be slightly higher trophic position, whereas benthic prey tend to be primary consumers. We'll come back to this slide a little bit later. Now, this variation in isotope signature indicating differences in diet is correlated with a number of morphological traits, most strongly the length of gill rakers. These are sieve-like structures in the mouth of the fish that filter food as the fish gulps water in. And if you have short gill rakers, you tend to eat more benthic prey. If you have long gill rakers, you tend to eat more limnetic prey, and we see this both using carbon and nitrogen isotopes. We see it by looking in stomach contents as well. If you have long gill rakers, you're eating more limnetic prey. If you have short gill rakers, you're eating more benthic prey. And I could show you lots of different prey taxa where we see these trends. Now, all of these data I've shown you are from individual lakes, fish that are caught on a single day. So this heterogeneity among individuals is standing between individual differences. And so the punchline here is that any single solitary population harbors quantitative variation in morphology, and at the ends of this distribution, individuals are feeding on fundamentally different foods, and that shows up in their stomachs and in their isotopes, both of which are correlated with each other and morphology. So then the next question is, are those morphological traits associated with diet variation subject to disruptive selection? Now, this is some older data, but we can move in and catch a bunch of fish and measure any of a number of fitness proxies. We've used relative growth rates using a biochemical assay with RNA-DNA ratio. We've used body size. We've used investment in reproductive tissue. And all of those repeatedly give us the trend where phenotypically average individuals have, on average, slightly lower fitness proxies than individuals at either tail of the phenotype distribution. And using cubic spline, using quadratic regression, we systematically recover this quadratic fitness landscape indicative of disruptive selection. So we can measure this by quantifying the curvature of this fitness landscape. If it's a positive curvature, a positive quadratic coefficient, then it's disruptive. If it's a negative curvature, then it is tending towards stabilizing. And so we can experimentally manipulate density of fish in enclosures using 150 square meter cages. And we find that at high density, when we basically double the number of fish per unit area, we see systematically positive quadratic curvatures indicating disruptive selection. In those same lakes, these are paired designs. Each pair of points is a different lake. At low density, we see stabilizing selection instead. So there's a systematic tendency for competition for food to generate disruptive selection. Now, I presented this here in Vienna at a meeting in 2006, and several theoreticians said, well, that's great, but is it common in nature? And I said, I will tell you in 3 years, and sure enough, 3 years later, we reported a survey of 15 populations looking at standing differences in fitness landscape. So in each of these populations, we sampled 400 or 500 individuals, generating a quadratic curvature. And each point here is a different lake. This slide represents 3 years worth of full-time work by a technician. She hates this slide. She hates the fact that it's summarized in one slide, 3 years of her life. But here you go. So this axis is the quadratic selection gradient, so disruptive is positive, neutral is in blue, stabilizing is in red, and you see that there's a systematic tendency towards positive curvatures. So disruptive selection, while not universal and not particularly strong, is in fact the dominant mode of speciation in these solitary populations of stickleback. So, seems like maybe they should be speciating. I had many people, when I first presented this stuff a number of years ago, say, well, if there's disruptive selection, why aren't they speciating? But of course, everyone in the room here knows that you need assortative mating also. Disruptive selection is not sufficient. So is there assortative mating? Well, we started to think that maybe that's possible, and that was based on a paper published by Ashley Ward. And what he did is feed a focal individual a food item, let's say Daphnia, for 2 weeks, and then put this fish in a tank with a tube at either end, and at this end the tube had fish fed Daphnia as well, and this tube had fish fed a different food item. And Ward found that the fish would preferentially associate, this was a shoaling decision, not a mating decision, would associate with the group of fish that had been feeding on the same food. And that was true as long as there were olfactory cues. And it was symmetric. If they switched this focal food item, they got this fish going to the other group. So there seems to be social decisions being mediated by olfactory cues derived from shared food experience. We call it the garlic eater's hypothesis. If you like garlic, maybe your mate likes garlic as well. Okay, so how can we test whether that's happening in these solitary populations? Let's go back to this graph I showed you earlier, where we have this axis of isotopic variation ranging from limnetic foragers to benthic foragers. If there's assortative mating, we should find that individuals at this tail of the ecological axis should be mating with each other. And individuals at this tail should be mating with each other. So we should see a positive correlation between the isotope signatures of males and females who have mated. So these positive correlations are a product of assortative mating. And so you can use the correlation coefficient between mated pairs as a measure of assortative mating on a basis of a shared trait. Now, unfortunately, it's very difficult to sample males and females as they mate in nature. They're pretty quick about it. It's hard to catch the females. But the nice trick is that both males and females vary along this axis, and females give their eggs their isotopes. So we can use the eggs as a proxy. A female comes in, she'll lay eggs in the male's nest and take off. So we can't get the female, but we can get her eggs. And it turns out that there's a very nice correlation between the female's eggs and the female's isotopes. This correlation is using muscle tissue from the female. We actually have a much higher R-squared if we use liver tissue because the lipid content is more comparable. So what we can do is spend more time snorkeling in frigid water. We collect males by dip net. We collect eggs from their nests. And we can ask, is there a male-egg correlation? We can sample females and take eggs from their ovaries and ask, is there an egg-female correlation? And we can use that to estimate the male-female correlation. And sure enough, when we measure isotopes, and I'm using, remember the nitrogen and benthic axis, there was some correlation there. So we're actually using the principal component of that isotopic axis here. And so we find that male signature and egg isotope signature are positively correlated, significantly so, indicating that there's likely some assortative mating on the basis of diet or some correlated trait, maybe morphology, for example. And we estimate a male-female correlation to be about 0.5. I think it was 0.49. So that's all very nice. So we have assortative mating. Now maybe it's just an artifact of spatial heterogeneity. So to test that, my student Lisa Snowberg, whose work this is, sampled spatially explicit nests where we used a very precise GPS, accurate to about 20 centimeters, to locate each nest. These dots are individual nests along a lake shore. And then we can ask whether there's a spatial gradient. So are the two shorelines different? Is there a spatial gradient in isotopes along a shoreline? And does isotopes vary by depth where the nests are? Turns out that there's some significant differences between the shores. There's no effect of axis along the shoreline. And there's also a depth effect. So males who are nesting deeper have a different isotope signature than males that are nesting shallower. Now, this is remarkable. This is one population. And what I'm telling you is that a difference of 1 meter depth is enough to explain differences in male isotope signatures. Male morphology also differs systematically, as do egg isotopes by depth. So there's some remarkably fine-scale structuring of where individuals are choosing to settle in their environment. And that could generate this male-female correlation, or male-egg correlation, if there are shared habitat preferences going on. Well, so we can take account of this spatial variation and take the residuals on these spatial trends, and we still find a female, or egg I should say, correlation with male isotope signature after statistically removing the effect of this spatial heterogeneity. And so, yes, there's a component of spatial structure generating this assortment, but there's additional assortment going on for some other reason as yet unidentified mechanistically. So to summarize the first half of this talk, we have ecological heterogeneity in these solitary populations, we have disruptive selection, and we have assortative mating. So great, shouldn't sympatric speciation be happening? Before I say no, let me point out to you that these are actually very general phenomena. So Kingsolver et al. In a 2001 review paper summarized the distribution of selection strengths, both linear and quadratic selection gradients, and they found, to many people's surprise, that positive and negative quadratic curvatures were about equally common and about equally strong. Selection is typically fairly weak, and in fact what we see in stickleback is about average quadratic selection gradients. But nevertheless, disruptive selection is, maybe a little surprisingly, moderately common even though it's weak. Same thing goes for positive assortative mating. A graduate student of mine, in collaboration with Mark Kirkpatrick and I, has done a meta-analysis of assortative mating where we use correlation coefficients between mated pairs in natural populations to generate a distribution of assortative mating strengths within populations. We're excluding species pairs, we're excluding incipient species, we're excluding host races, we're excluding discrete morphs. This is within population assortment. And what we found, much to our surprise, is a systematic tendency towards positive assortative mating. This is from just over 1,000 estimates of correlation coefficients from over 200 species from 5 phyla. The average strength of assortment is about 0.27, 0.28, which is not strong, but it's systematically different from 0. So the idea that disruptive selection and positive assortative mating are widespread phenomena is quite reasonable. So that's good news for the theoreticians who've been assuming both of these processes all along. Of course, there's the caveat. Why do we not see speciation? Well, one possibility is that although these processes are present, they may not be strong enough. So for example, in this distribution of assortative mating strengths that I just showed you here, stickleback are slightly more assortative than average. Remember, there's a correlation coefficient of about 0.5 in one of our estimates, whereas the average across all of the organisms we see is about 0.27. Notably, fish as a group are more assortative than the other clades that we subdivided our data set into. Now, just as an aside, to the theoreticians in the audience, this should hopefully be useful information in the sense that starting conditions in populations may typically not be strictly random starting conditions. So why do we not see speciation? Well, maybe there are some reasons. So earlier this week, I just ran some quick and dirty simulations. I will admit that these were just stochastic, numerical, individual-based simulations where I generated a distribution of phenotypes using a multi-locus additive model. I applied disruptive selection of a specified strength. I applied assortative mating of a specified strength. And I just asked, do I see the emergence of a bimodal, strongly bimodal distribution of phenotypes? And the answer is sometimes you do in a subset of parameter space. And then I mapped on where stickleback are in this parameter space. So this is the average strength of disruptive selection and assortative mating in stickleback. These bars represent the range, and I've cut it off. Remember, there were some negative quadratic gradients, so this bar on the selection axis would extend to be negative as well. It's not really relevant for the sympatric speciation question. And so stickleback basically are going to be in an oval right here, which obviously does not fall into the area where, at least in my version of these simulations, we were seeing sympatric speciation. And I certainly invite the theoreticians in the audience to help me do a better job of this particular task and ask, why is it do these solitary populations not speciate? Maybe one answer is that they shouldn't. These processes just are not strong enough. Now, of course, if we have disruptive selection, maybe we should see over time some reinforcement, the evolution of increased assortative mating, and maybe that would bring some of these populations that are under stronger disruptive selection into an area where speciation is possible. So we might potentially evolve in that direction and generate speciation, but maybe not. Let me give you a few reasons why that might not happen. One is that these selection gradients are not fixed. So for example, First Lake in 2005 was one of the populations where we saw the strongest disruptive selection in that 15-lake comparative study. Well, we went back in 2008, used exactly the same methodologies, sampled in the same way, same time of year, and we see significant stabilizing selection. Another lake that had the second strongest disruptive selection in 2005. In 2008, we saw directional selection. So the fitness landscapes may be fluctuating. Why? Well, I showed you earlier that fitness landscapes are density dependent. Stickleback go through variations in population size that might toggle back and forth between disruptive and stabilizing selection. Another possibility is that not all lakes are systematically likely to be subject to disruptive selection. Remember this slide earlier, there's a lot of variation here. Yes, on average, quadratic selection is disruptive, but we have counterexamples in individual lakes. Well, why is that? One possibility has to do with the ecology of this lake versus these lakes. Well, what about the ecology? Well, ecological opportunity may dictate the fitness landscape such that very small lakes are predominantly benthic habitat with proportionally little limnetic. Large lakes are predominantly open water with proportionally little benthic habitat. And it's really these intermediate lakes that have a mixture in some approximately equal ratio of carrying capacities where we expect that disruptive selection to be most pronounced. And sure enough, we have a significant quadratic relationship between the ratio of littoral to pelagic habitat and the strength of quadratic selection, such that disruptive selection is typically only seen in these intermediate-sized lakes, and we're seeing neutral or stabilizing selection at the extremes of this lake axis. So maybe only some lakes are candidates to begin with. And even, I should point out, even there, we're still seeing some years with and some years without disruptive selection. Okay, another possibility is that there are alternative outcomes to this disruptive selection. Just because you have disruptive selection doesn't mean speciation is the only option to alleviate the inequality of the strength of competition. So for example, Michael Dobley and I, a number of years ago, generated a paper showing that the theoretical conditions for ecological sexual dimorphism, character displacement between the sexes to mitigate competition, the conditions were the same as the conditions for sympatric speciation. So this is a case where we start with a population and it evolves towards a carrying capacity at that horizontal line. As it approaches the carrying capacity, it comes under disruptive selection. It then bifurcates as long as you have some independent control of the male and female phenotypes. It can bifurcate to generate a sexual dimorphism. Well, what do you know? When we look at the strength of sexual dimorphism in these populations, it's maximized at the same lakes where we expect to see the most disruptive selection. And we see relatively little sexual dimorphism in the big limnetic lakes or in the smaller, more benthic lakes. And again, this is a significant hump-shaped relationship here. Now, there's a corollary to this. When you evolve this dimorphism, it evens the strength of competition across the range of resources, and so the fitness landscape should flatten a little bit, or maybe completely. So as a consequence, there's a prediction from this model that the strength of dimorphism should, all else be equal, as dimorphism goes up, disruptive selection should go down. So we'll statistically control for lake size now. We'll look at the residuals of dimorphism with lake size and the residuals of disruptive selection with lake size. And we see that for two different phenotypic characters, there's a negative correlation, although in one case it's not significant, a negative correlation between the strength of disruptive selection and the strength of dimorphism. When dimorphism evolves, disruptive selection gets weaker. Now that's important because, at least in our models, and Jenny Bauman has some similar results, dimorphism tends to happen faster than speciation. It mitigates the strength of disruptive selection, which may drive populations away from that area in parameter space where speciation is plausible. So the fast evolution of dimorphism may make sympatric speciation less likely. Now, dimorphism is only one option. You can also evolve increased variance, and Daniel Berner and I have a paper that just came out in Evolution showing that when you move from a marine ancestor to fresh water, you have both rotation of the phenotypic covariance matrix, and you also have elongation. So we see the evolution of increased phenotypic variability in these freshwater lakes. And we can use eigentensor analysis to summarize variation in these p-matrix size and orientation. And the first eigentensor that explains the most difference between lakes in their p-matrix basically is differences in variance, elongation. And it's correlated, what do you know, with lake ecology. Different lakes ecologically have different degrees of variability. I would have predicted, again, a hump-shaped relationship. That's not what we see. We see actually just a linear relationship, although with increasing noise as you go to this end, and I can't tell you much more about why that is yet, but it's an intriguing trend. There's also behavioral diversification. Richard Svanbach and I did an experiment in which we manipulated density over a 2-week period in field enclosures, and then we looked at how heterogeneous individual stomach contents were, how variable the 30 fish were in this enclosure or that enclosure. And we see that between individual variability was higher at high density than low density. There was no effective mortality. There was no phenotypic plasticity over this time scale. Instead, the correlation between morphology and diet got stronger because individuals were behaviorally maximizing their feeding efficiency on the basis of their morphology. So to summarize, Sympatric speciation, I think many people would agree at this point, is now widely regarded as, quote, theoretically plausible. And I don't quite know what theoretically plausible means other than that you can write down equations or run simulations that will cause it to happen. I think the bigger question is how biologically plausible the theoretical assumptions are. And for that, we need these conversations between theoreticians and biologists. And I think the punchline that I have for you today is that the key assumptions are right qualitatively, but maybe wrong quantitatively to get speciation. It's still not that easy. Thank you.
Thank you very much, Jen. Very nice talk, and thank you for speaking so well at this time. And we have questions. Somebody needs a microphone to the end.
In lakes with and without species pairs, what does the molecular genetics tell you?
What does
The molecular genetics tell you?
So in the species pair lakes, the genetics are ambiguous about the origins of those species pairs. Depending on which locus you look at, you get a different answer. Although typically for nuclear loci, it seems like they were independently derived, and the limnetics are maybe more closely related to marine than they are to their fellow benthics. In freshwater, for all of the microsatellites we've looked at for about 20-odd populations, it's Hardy-Weinberg equilibrium across the board.
Next question, Lisa.
I would like to ask how old your system is because it may just take time.
Yeah, so the system, the lakes range from, well, the lakes are all about 12,000 years old. Not all lakes were colonized instantaneously. We have evidence that there's a progression as you move up a watershed. It's more and more recent colonization. There's a gradual stepping stone process. I've used the empirical estimates, the strongest disruptive selection combined with the strongest assortative mating that I've seen in stickleback, and I've run those simulations for 200,000 generations, and I still didn't see any speciation. So maybe if I ran it for 10 million, I don't know, but yes, of course it's possible that if we just waited long enough, they would all split. But on the other hand, given the fluctuating selection gradients, the alternative outcomes to speciation, I'm fairly comfortable saying that these things probably won't speciate typically, especially given that the species pairs don't have particularly clear evidence of sympatric divergence.
I already see 3 more hands. Let's find us over there in the back.
Thank you for your response. My question is related to you mentioned ecology a few times, and both the parameters you mentioned are abiotic. And the perspective that you talk about is all from the fish. What do you think is going on with the prey populations and driving selection differences or the way that maybe fluctuate between those two different parts?
It's a great question. So the question was, what role do the population and community dynamics of the prey play in generating selection? I don't think we have a great idea about that. I mean, I can tell you that when we increase density, we get more disruptive selection, and it coincides with reduced prey availability. We've done some experiments that are more community ecology, looking at how the strength of trophic cascades induced by stickleback changes as the variance of the phenotype distribution. So we manipulate the phenotypic variance and ask whether that alters the community dynamics. But at this point, we really don't have a good idea. And that gets at this issue of ecoevolutionary feedbacks. And I think the big problem is, so all of my experiments are in the field, and it's very hard to effectively manipulate prey distributions in the field.
Then you commit to the answer as to why, as to whether or not the limnatic and species. And if so, why?
I do my best to avoid species delimitation tasks. I think that given the low rate of hybrids in nature and the various layers of pre- and post-mating reproductive isolation, I have no problem considering them biological species. I'm not one to say that biological species concept requires a hybridization rate of exactly 0, nor am I one to say that a set of species have to be permanent in the sense of being irreversible. So yeah, I'm content with considering them distinct species. They've never been described as such in the sense that nobody's ever given them distinct Latin binomials.
Okay, next question is over here.
Approach to test for the likelihood of sympatric speciation going on in various species. But to make it a bit more general, for example, if you want to include seedlings who also have sexual selection as one of the potential driving forces, would it be possible to include sexual selection into this kind of approach?
Would it be yes, I suppose in principle it would be possible to include sexual selection into this approach. Interestingly, Jenny Bauman has data suggesting that patterns of sexual selection within the benthic and limnetic species do not extrapolate to explain the reproductive isolation between the species. So for example, although body size is a major component of species distinction by the fish, it's not subject to sexual selection within the populations. So, but I suppose in principle it's the case, but I think I would argue to the audience here that what we need are sets of theories that are couched in parameters that I can go out and measure. So I need to know what is it that I can go out and experimentally or observationally quantify that you need to know.
Okay, we have 2 more questions. The first is by Jürgen. So, just one simple question. What if you did the experiment of, not saying you should, but if you did it, of introducing marine sticklebacks into some of this lake every one at one time, would you expect speciation or just simple character of this pattern distribution?
That's a great question. So, I don't know. You know, the argument would be that if you have a double colonization event, if you went to the more benthic lakes and you introduced a rather limnetic-like marine population, that yeah, maybe you would get some reinforcement and some ecological character displacement. Getting permission to do that, of course, is challenging. Now, I don't think it's necessarily impossible, and I think if there was enough motivation by a big enough community of people, there are enough lakes with solitary stickleback that managers and conservationists might be convinced to allow such a thing. So for example, Michael Bell has been working with people in Alaska that are using rotenone to kill off all of the trout in a given lake because pike have been introduced. So there are these invasive fish in lakes in Alaska. They're tearing up the ecosystem, so they use rotenone to kill every fish in the lake. So then Mike Bell says, well, this is an opportunity. Can you let us reintroduce marine fish into these lakes? And they have replicate lakes, so they're going to be able to track the evolutionary trajectory of adaptation to fresh water. Now, all we need to do is do that in lakes where they haven't killed off all of the residents, and there you go.
Okay, one last question like this, please.
Okay, I'm thinking about those thousands of lakes where you just have one species. But anybody has an idea how those populations are regulated? Whether they are regulated by predators, by parasites, because if they are not regulated by food, of course.
So, many of the lakes do contain predators. The major predators are trout, diving water birds like loons and grebes, and some insects. But there's still not a clear data. I can't point to any study that has tried to quantify various forms of ecological regulation. I can say that when we experimentally reduce density in enclosures in nature, the fish's growth rate goes up, their investment in reproductive tissue goes up. When we increase density, both of those parameters go down. So that it does seem like there is some constraint on growth and fecundity being generated by food availability. because infection rates don't change when we do that, and they're predator-free cages. So I think that there's certainly at least some component that's a bottom-up density regulation from food availability. Parasite loads are hugely variable from lake to lake. We have lakes where, and this is one of the major directions in my research now, we have lakes where there are 90% of fish are infected with these schistocephalus that are these cestodes that weigh 2/3 the body mass of the fish. And there are other lakes a kilometer away that have zero infection. Yeah.